So, hello students, I hope all of you are doing great. Welcome back to your own channel, Vedantu Science Olympiad, and this is Basavaraj, so your biology master students, master teacher. Welcome back, all of you students. Welcome back. हमने पिछले क्लास में पूरा का पूरा लाइट रिएक्शन था ना, जो साइक्लिक और नॉन साइक्लिक फिनिश कर दिया था. So, if you have not watched the previous lecture, please do right now. So, go after the class, watch the first part because that is very important. Now, students, can anyone remind me in light reaction? What are the products in light reaction? Anyone, students? Anyone who can remind me what are the products in your light reaction? Cyclic या non-cyclic? हमारे non-cyclic के अंदर are we getting ATP? Yes, we are getting ATP. Are we getting something called as your NADPH? Yes, we are getting NADPH. Now, students, listen to me. All of you listen to me very carefully, students. Light reaction के अंदर है ना, light reaction के अंदर जो ATP और NADPH बन रहा है ना, ATP और NADPH, वो ATP और वही NADH, it will be used in your dark reaction. That ATP and NADPH will be used in your dark reaction. It will be used, and with the help of that usage, what will be made? C6H12O6. Glucose will be made using the ATP and NADPH from a light reaction. Now, students, in today's class, we will be focusing on the entire dark reaction. The dark reaction is also called as light independent reaction. What? Light independent reaction. It is called as light independent reaction because it does not require light. Light की जरूरत नहीं है, students. Light की जरूरत नहीं है. Okay. All of you students, hello. Students, what happened? Hello, students, what happened? All of you. Chill. Okay. Now, chill, students. So, whatever used in your light reaction is used in your dark reaction. Okay. Now, first thing which you need to understand about dark reaction is that it does not require light. Perfect, students. Then, requires the products of a light reaction, NADPH and ATP. Yes. It is also called as biosynthetic phase. Why? It is called as biosynthetic phase because there is synthesis of glucose is happening in this reaction itself. Okay, synthesis of glucose is happening in this reaction itself. So if you ask me, students, if you ask me what are the raw material to make glucose, can anyone tell me, students, what are the raw materials required? The raw materials required are ATP, NADPH and CO2. These three are the raw material which are very important to make glucose. अगर ये तीनों के तीनों नहीं है, डार्क रिएक्शन नहीं होगा. अगर डार्क रिएक्शन करना है तो ये तीनों की जरूरत है. ATP and NADPH, ये दोनों की जरूरत कहां से आ रहे हैं? Both of these are coming from the light reaction, and CO2 it is coming from the atmosphere. Okay? So when all of these three are entering the dark reaction, glucose is finally prepared. Okay? Students, शुरू करें अभी? शुरू करें क्या? Okay. Now, all of you know, light reaction happens on the thylakoid, it happens on the grana, it happens on the stromal lamellae. But where does the dark reaction happens? Does anyone know, students? Does anyone know where does the entire dark reaction takes place? What is the location of dark reaction? So please remember, students, the entire dark reaction takes place in the case of stroma. In the case of stroma, it happens in the case of the matrix or the stroma. Okay, done, students, done. Now here is your entire dark reaction on the board. पूरा का पूरा dark reaction हमारे board के ऊपर है. Now, students, okay. Now let me just go one by one. Every single step. Now, before the class, I've written it for you for it be easier to understand. Now, in this entire dark reaction, students, it is also called as Kelvin cycle. The dark reaction is also called as Kelvin cycle. Why? Because it is called as Kelvin cycle based on the discovery of the scientist. The scientist who discovered this, right? His name was Melvin Kelvin. Melvin Kelvin नाम था उनका. So, because of the scientist, we have the Kelvin cycle name right now. Okay. Now, what is the first step? First step is very simple, students. The first step is very simple. First step में क्या हो रहा है? First step में the molecule called as RUBP, ribulose 1,5 bisphosphate. What is this? Ribulose 1,5 bisphosphate, which is a ketone sugar. It is a ketone sugar. Now this ketone sugar will accept a CO2 molecule. It will accept a CO2 molecule. So in the first step, 5 carbon RUBP plus 1 carbon CO2. Both of these will combine. In this entire reaction, there is an enzyme involved. What happens, students? There is a enzyme involved here. The enzyme is called as what? Rubisco enzyme. Rubisco enzyme is the most abundant. हमारे अर्थ के अंदर ना सबसे ज्यादा यही एंजाइम है स्टूडेंट. The most abundant enzyme here. Okay, the most abundant enzyme here. So the first step is called as what? The first step is called as carboxylation step. The first step is called as what? Carboxylation step done. Point number 1. In carboxylation step, RUBP and CO2 are combining to give you something called as PGA. Now what is PGA, students? 3-phosphoglyceric acid. What, students? 3-phosphoglyceric acid. Now please remember, this 3-PGA is actually 3 carbon. So this is 3 carbon. Here we have 5 plus 1, 6 carbon. How does it match? Does it match? यहाँ पर 3 कार्बन का PGA, यहाँ पर 5 कार्बन जुड़ रहे हैं यहाँ पर. So what do we do, students? What do we do in that case? In that case, we get 2 molecules of 3-PGA. So 3 into 2 is 6 carbon. 5 plus 1 is 6 carbon. So 6 carbon is taraf, 6 carbon is taraf, balance is maintained. Okay. So in the initial stage, in carboxylation, our enzyme is called as Rubisco enzyme. RUBP plus CO2, we will get PGA molecule. Now students, now पहला step हो गया students, पहला step हो गया, खत्म हो गया यहीं पर. Now in second step, what happens? In the second step, called as reduction stage, the PGA gets converted to triose phosphate. In the reduction stage. Now this is where यहीं पर ना जो ATP और NADPH बने थे ना लाइट रिएक्शन के अंदर, उसका यूटिलाइजेशन यहीं पर होगा. अभी यहीं पर होगा स्टूडेंट्स. ठीक है, देखो यहां पर इन दिस रिडक्शन स्टेज द 2 NADPH मॉलिक्यूल्स, कितने स्टूडेंट्स? 2 मॉलिक्यूल ऑफ NADPH, 2 मॉलिक्यूल ऑफ ATP are getting completely consumed right now in the reduction stage. So 2 NADPH consume हो गए यहां पर और 2 ATP भी consume हो गया, अभी consume पूरे. And with the help of this entire reduction stage, we get triose phosphate, which is 3 carbon. 3 carbon, 3 carbon, same thing. Now, this triose phosphate will eventually convert into glucose. This will get converted to glucose eventually. It will get converted. Okay? Now what happens in the next step? The next step is called as regeneration. In regeneration, what happens, students? Triose phosphate again converts back to RUBP. Regeneration is happening. And in this regeneration stage, again, 1 ATP is consumed. So in reduction stage, 2 ATP and 2 NADPH, and in the regeneration step, 1 ATP is consumed. So can anyone tell me, in one dark reaction, how many ATP and how many NADPH is consumed for 1 CO2? Anyone student? This is for 1 CO2. This entire cycle here, students, this entire cycle here is for 1 एक ही एक CO2 के लिए. So can anyone tell me, students, how many ATP and how many NADH? Quick. For one cycle, in one cycle, for 1 CO2, we in total, for 1 CO2, to fix 1 CO2, we require 3 ATP and 2 NADPH for 1 CO2. एक CO2 के लिए. But students, in the case of glucose, how many carbon do we have, students, in glucose? In the case of glucose, all of you know the formula. It's C6H12O6. In the case of your glucose, we have 6 carbon. We have how many carbon? 6 carbon. And this cycle here is for 1 carbon. So to make glucose which has 6 carbon, does that mean this cycle has to happen 6 times for every single carbon? देखो स्टूडेंट्स, हर एक कार्बन बनाने के लिए, यहां पर हर एक कार्बन बनाने के लिए द एंटायर साइकिल हैज टू गो थ्रू 6 टाइम्स, क्योंकि this is one cycle for one carbon, but we need how many carbons in glucose? 6 carbons. So the entire cycle has to go 6 times to make one carbon molecule. Now let's see the energy, students. Energy कितना मिलेगा यहाँ पर? See, 3 ATP for one carbon. For 6 carbon, how many ATP? That is 3 ATP into 6, 18 ATP, है ना? 18 ATP बन गया. Now 2 NADPH, right, into 6, that is 12 NADPH. So we require 12 NADPH and 18 ATP to make one glucose molecule. To make one glucose molecule, एक ग्लूकोज मॉलिक्यूल बनाने के लिए इतना ATP और इतना NADPH की जरूरत है स्टूडेंट्स, एक जरूरत है. ओके, सो इतना जरूरत है. Clear students? Clear all of you? Clear? Perfect students. This is called as C3 cycle also. Now you might be wondering, sir, why is it called a C3 cycle? It is called a C3 cycle because the first stable product here, is actually 3 carbon here, 3 carbon. That is why it's called a C3 cycle. 3 C3 cycle. Okay, C3 cycle. Okay, done students, done. Now here are a few examples: wheat, soybean, oats. 3 different examples of your C3 plants. Students. Now what is photorespiration students? इसका मतलब क्या है? Students, very simple logic. हमारा जो यहाँ पर एंजाइम है ना, एंजाइम रुबिस्को एंजाइम, ये देखो, ये जो रुबिस्को एंजाइम है ना, रुबिलोस 1,5 कार्बोक्सिलेज ऑक्सीजनेस. This Rubisco enzyme is very what do I say, very cunning. जिसका बर्थडे क्लोज है उसके साथ फ्रेंडशिप बढ़ जाती है इसकी. If this particular Rubisco enzyme, it can bind with CO2, which is very good. This Rubisco enzyme can bind with CO2 and help in C3 cycle. But the same Rubisco can also bind and work with oxygen also. Oxygen के साथ भी कर सकता है students. It's like I will do with both. Whenever there is oxygen के पास. Whenever there is CO2 के पास. Now what are the conditions जहां पर ना ये Rubisco oxygen से कब bind करेगा? That is very important students. That is very important. Okay. To understand, let's understand here. जब भी एटमॉस्फेयर में CO ऑक्सीजन बढ़ जाएगा, ना, the Rubisco will go and bind with oxygen. It will leave CO2 behind. CO2 को बोल देगा टाटा बाय बाय, चलता हूं मैं. Okay, so Rubisco will bind with oxygen and that can cause photorespiration. Or whenever the CO2 concentration is very low, छोड़ देगा CO2 को, CO2 का साथ छोड़ देगा. हो छोड़ दिया स्टूडेंट्स. Then whenever there is high light and whenever there is high temperature, okay, whenever there is high temperature, photorespiration will happen. Now what is photorespiration students? What is photorespiration? In the case of photorespiration, what is happening is the RUBP is forming a bond with oxygen right now because of this enzyme right here, this enzyme, Rubisco enzyme. Because of this enzyme, in this condition, RUBP is forming a bond with oxygen right now. Now when this bond happens, what happens? We get something called as PGA and we also get something called as PG. Now what is PG? Phosphoglycolate. देखो स्टूडेंट्स, यहाँ पर 5 कार्बन, यहाँ पर 0 कार्बन्स, so 3 plus 2 is 5 कार्बन. Now this is called as a very wasteful process. बहुत ही वेस्ट है ये प्लांट के लिए. क्यों? क्योंकि इसके अंदर ना ही एनर्जी बन रही है, ना ही कुछ ग्लूकोज बन रहा है अच्छी तरह से, कुछ भी नहीं हो रहा है. एनर्जी CO2 ऐसे वेस्ट निकल के जा रहा है. यहाँ पर there is no sugar formation, ATP is used, CO2 is released outside, NADPH नहीं बन रहा, कुछ भी नहीं बन रहा. सो प्लांट्स के लिए सबसे वेस्ट है ये प्रोसेस, सबसे वेस्टफुल प्रोसेस है, इट्स आल्सो कॉल्ड एज अ वेस्टफुल प्रोसेस, क्योंकि इसके अंदर ना कुछ जरूरत की चीज कुछ नहीं बन रही इसके अंदर, सिर्फ घाटा हो रहा है हम पर सब कुछ. Okay, that is what is happening in the case of photorespiration. So photorespiration is actually very bad for the plants, and this happens because of the rubisco is helping to form a bond with the oxygen right now. That is called as photorespiration. Now this is the actual photorespiration. It happens in three different organelles. Chloroplast में थोड़ा थोड़ा होगा, peroxisome में थोड़ा थोड़ा होगा, फिर mitochondria में थोड़ा थोड़ा होगा. So this is the actual cycle. इतना डिटेल में जाने की जरूरत नहीं है. यहां पर यू हैव टू ओनली रिमेंबर द थ्री डिफरेंट ऑर्गेनल्स. कौन कौन सा स्टूडेंट्स? क्लोरोप्लास्ट, पेरोक्सिसोम, माइटोकॉन्ड्रिया. थ्री ऑर्गेनल्स इन्वॉल्वड इन फोटोरेस्पिरेशन, एंड फोटोरेस्पिरेशन इज वेरी बैड फॉर द प्लांट्स. बहुत ही जरूरत नहीं है उसकी. ओके, नाउ स्टूडेंट्स, नाउ डू यू थिंक देयर आर सम प्लांट्स जो वेरी स्मार्ट, देयर आर सम प्लांट्स जो वेरी स्मार्ट जिसके अंदर फोटोरेस्पिरेशन होता ही नहीं है. Students, do you think there are plants like that? Do you think there are plants like that जिसके अंदर photorespiration होता ही नहीं है? है students, बहुत है. We call them C4 plants. ये smart क्यों है students? Smart क्यों है? सोचो, ये plants बहुत smart है क्योंकि अगर हम एक कमरा बनाएंगे इस तरह से, एक कमरा बना देंगे, इसके अंदर एक कमरा, इस कमरे के अंदर हम CO2 को ट्रैप कर देंगे. क्या करेंगे? इस कमरे के अंदर CO2 को ट्रैप कर देंगे, ट्रैप. और इसी कमरे के अंदर रुबिस्को भी होगा, रुबिस्को भी होगा. नाउ टेल मी स्टूडेंट्स, अगर इस कमरे के अंदर, बंद कमरे के अंदर रुबिस्को भी है और CO2 भी है, और इस कमरे के अंदर ना, इस कमरे के अंदर ऑक्सीजन का एंट्री हो ही नहीं सकता, ऑक्सीजन का एंट्री हो ही नहीं सकता. कंप्लीटली शट आउट, कंप्लीटली शट आउट. Now you tell me students, इस कंडीशन के अंदर फोटो रेस्पिरेशन हो सकता है क्या? नहीं हो सकता, क्योंकि इस कमरे के अंदर अभी सिर्फ CO2 है और उसके साथ रुबिस्को एंजाइम है और RUBP और सब कुछ है इसके अंदर, बट ऑक्सीजन की एंट्री नहीं होगी. This is what is happening in the case of C4 plant. In the case of C4 plant, the C4 plants are trapping the CO2 and rubisco enzyme in one special type of cell. Students, ready to learn? All of you ready for C4 cycle? All of you ready for C4 cycle, students? Ready for C4 cycle? Yes? Ready? All of you ready for C4 cycle? Okay, students. Now, this is the anatomy of C4 cycle. The anatomy of C4 cycle is very special. It's very different. The anatomy is called as Kranz anatomy. The anatomy is called as what? Kranz anatomy in the case of C4 plant. अगर यहाँ पर दिख रहा है आपको स्टूडेंट, यहाँ पर दिख रहा होगा, here all of you notice very carefully. Now here can all of you notice, बाहर की तरफ we have a lot of mesophyll cells, a lot of mesophyll cells on the outside. इस mesophyll cell के अंदर CO2 घुस रहा है, बट अंदर की तरफ क्या स्टूडेंट्स? अंदर की तरफ बहुत सारे tightly packed in a wreath like structure. इसको हम कहते हैं, क्या कहते हैं इसको हम कहते हैं wreath like structure. दिख रहा है ये वाला? It's called as wreath like structure. इस wreath like structure के अंदर ना, इस wreath like structure के अंदर ऑक्सीजन की एंट्री कभी नहीं हो सकती. इस wreath like structure के अंदर ऑक्सीजन की एंट्री कभी नहीं हो सकती इसके अंदर. So this entire structure here is called as the it's called as bundle sheath cells. They're called as what? They are called as bundle sheath cells. और इस bundle sheath के cells के अंदर कुछ ऑक्सीजन नहीं घुस सकता. Now students, so see this again, you get one more idea. बाहर की तरफ क्या है स्टूडेंट्स? बाहर की तरफ है मेसोफिल, बाहर की तरफ है मेसोफिल सेल. ओके, अंदर की तरफ क्या है? अंदर की तरफ टाइटली पैक्ड बंडल शीट सेल्स, टाइटली पैक्ड बंडल शीट सेल्स अंदर की तरफ है. इट इज इन अ wreath like structure. It's called, this is called as a wreath. देखा होगा ना टीवी के अंदर, द क्रिसमस के अंदर सब कुछ देखो, दिस इज कॉल्ड एज wreath. दिख रहा है इस तरह, सेम इस तरह देखो, wreath like structure here, made up of bundles, made up of bundle sheath cells. These are called as bundle sheath cells. दिख रहा है ना bundle sheath cells. Now students, now let's understand what exactly happens here. Bundle sheath cells are arranged in a form of a wreath around the vascular bundles. Vascular bundles के around we have bundle sheet cells. Okay. Students, I don't teach in any camps. Students, I teach for NEET students. मैं NEET पर students को पढ़ाता हूँ. I teach for second PUC. I teach for 12th board exams के लिए पढ़ाता हूँ. ओलंपियाड में पहली बार पढ़ा रहा हूँ मैं ओलंपियाड में, है ना. सो कैंप में नहीं, मैं नहीं हूँ कैंप के अंदर, ठीक है. Now, bundle sheet cells are characterized by having large number of chloroplasts, thick walls and no intercellular space. यहां पर ICS is completely absent. Intercellular space is completely absent here. So can any form of gas enter from outside here? Students, tell me, can any form of gas enter from outside to inside bundle sheet? नहीं. किसी भी प्रकार का बाहर से ना अंदर की तरफ कोई भी गैस नहीं आएगा, क्योंकि यहां पर सारे के सारे सेल्स बहुत ही टाइटली पैक्ड हो गए हैं बंडल शीट के अंदर. ठीक है, बंडल शीट वाले. नाउ स्टूडेंट्स, now this is also called as hatch and slack pathway. Hatch and slack pathway because the scientists who discovered this. Okay, now students, here is a structure of C4 cycle. Here is a structure of C4 cycle. अगर आप देखोगे ना, यहां पर बाहर की तरफ क्या है, बाहर की तरफ यहां पर मीसोफिल से बना हुआ है, मीसोफिल सेल है ना, मीसोफिल सेल. और अंदर की तरफ क्या है स्टूडेंट्स, अंदर की तरफ है बंडल शीट सेल्स. ये बंडल शीट सेल्स है ना, बहुत टाइटली पैक्ड है, बहुत टाइटली पैक्ड है. और इस बंडल शीट के सेल्स के अंदर, इस बंडल सेल्स के अंदर किसी भी प्रकार का बाहर से अंदर की तरफ कोई भी गैस नहीं आएगा. अभी बाहर से अंदर की तरफ कोई भी गैस नहीं आएगा बंडल शीट के अंदर. जो कुछ होना है अंदर की तरफ होना है. अभी देखते हैं स्टूडेंट, देखो पहला स्टेप क्या है स्टूडेंट. पहला स्टेप है यहां पर द एक्सेप्टिंग द CO2. यहां पर क्या है, यहां पर CO2 इज एक्सेप्टेड बाय PEP, phosphoenol pyruvate. सो CO2 इज एक्सेप्टेड बाय PEP हियर. पर यहां पर क्या हो रहा था स्टूडेंट्स, यहां पर क्या हो रहा था, हमारे इसको कॉपी कर लेते हैं, नहीं चिल्लाते हैं. यहां पर रूबिस को उठा रहा था ना, यहां पर Rubisco was inviting or welcoming. यहां पर Rubisco was welcoming the CO2. बट यहां पर कौन कर रहा है स्टूडेंट्स, यहां पर कर रहा है PEP, phosphoenol pyruvate. Now phosphoenol pyruvate is 3 carbon students. Phosphoenol pyruvate is 3 carbon. CO2 is 1 carbon. What is 3 plus 1? Students, what is 3 plus 1? Obviously 4. So here we get a first stable 4 carbon compound here. The first stable 4 carbon compound is called as oxaloacetic acid, OAA, oxaloacetic acid. And this OAA is 4 carbon. That is why it's called as C4 cycle. It's called as C4 cycle. Clear all of you? Clear? It is called as C4 cycle because of the first stable 4 carbon name. यहां पर वही था स्टूडेंट्स, यहां पर भी क्या हो रहा था, एक मिनट स्टूडेंट, एक मिनट. आवाज आ रही स्टूडेंट्स, आवाज आ रही है मेरी? हाँ. Okay students, now here if you remember students, here the first was 3 carbon compound, right, PGA. That is why it is called as what, C3 cycle. यहां पर क्या स्टूडेंट्स, यहां पर हमारा C4 cycle बना हुआ है, C4 cycle. Okay, now the enzyme involved is called as pepcase enzyme. Now what is pepcase enzyme? Pepcase enzyme is an enzyme, pep carboxylase enzyme, which is helping in the starting reaction here, pep carboxylase enzyme. This is most enough students. This is more than enough. Don't worry, sufficient. Now this OAA now converts to malic acid. Now, this malic acid is transferred to the bundle sheet. Transfer हो गया. Now this malic acid now converts to OAA again back, oxaloacetic acid. Now this OA, listen to me very carefully, students. Listen to me very carefully, friends. Listen to me very carefully here. Now this OAA now converts to something called as pyruvic acid. Pyruvic acid is 3 carbon. OAA is 4 carbon. So can I tell one CO2 is released outside here? One CO2 is released here. है ना. Once CO2 is released, you can see here, it is released here, CO2, and now this CO2 will enter the C3 cycle. So can I tell, students, can I tell C3 cycle is taking place in every single plant? It can be a C4 plant or C3 plant, पर दोनों के अंदर C3 cycle is still happening. Even though I call it a C4 cycle, even though I call it as C4 cycle, इस C4 cycle के अंदर C3 cycle चल रहा है अंदर की तरफ. देख रहे हो स्टूडेंट, देख रहे हो? सो अगर C3 cycle चल रहा है यहां पर ना, सो कैन आई टेल रूबिस्को एंजाइम होगा यहां पर अंदर की तरफ अभी. सो यहां पर क्या होगा, रूबिस्को एंजाइम भी पाया जाएगा, रूबिस्को एंजाइम भी पाया जाएगा इसके अंदर. सो क्रेजी कॉन्सेप्ट है ये. Okay, now this pyruvic acid is sent back again, it gets converts back to PEP regeneration step. Okay, so it happens, C3 cycle happens here also. Okay, so difference देखो. See, cell type in which Calvin cycle takes place. यहाँ पर Calvin cycle कहाँ पर हो रहा है, मेसोफिल, यहाँ पर बंडल शीट के अंदर. देखो हमारे C3 के अंदर सिर्फ एक ही सेल था, C3 के अंदर सिर्फ एक ही सेल था, यहाँ पर मेसोफिल सेल. पर यहां पर क्या है स्टूडेंट्स, यहां पर बंडल शीट और मेसोफिल दोनों यहां पर है ना, दोनों यहां पर. सो इसके अंदर क्या हो रहा है, इस बंडल शीट के अंदर यहां पर सीधे साइकिल बन रहा है. दैट्स व्हाट वी टोल्ड हियर. ओके, सेल टाइप इन विच इनिशियल कार्बोक्सिलेशन हैपेंस, मेसोफिल, दोनों में मेसोफिल. देख रहे हो यहां पर इनिशियल कहां पर हो रहा है, इनिशियल फिक्सेशन C3 में हो रहा है ना, C3 में हो रहा है स्टूडेंट्स. हाउ मेनी सेल टाइप्स, वन सेल, टू सेल्स हियर. प्राइमरी CO2 एक्सेप्टर RUBP, PEP. RUBP is 5 कार्बन, PEP is 3 कार्बन, है ना. नहीं स्टूडेंट, वन सेकंड, हाँ 3 कार्बन 5 कार्बन. ओके, देख रहे हैं यहां पर 3 कार्बन था PEP, यहां पर था ये 5 carbon RUBP, 5 carbon RUBP students. Okay, question practice I'll do later on students, we'll do it Tuesday. Okay, which is the primary CO2 fixation product, PGA we get here, which is 3 carbon. Here we get OAA, 4 carbon oxaloacetic acid, 3 phosphoglyceric acid. Okay, does the plant have rubisco? Yes, both have rubisco. पर यहां पर पेपकेस नहीं होता, सिर्फ यहां पर पेपकेस होता है, सिर्फ C फॉर्म में पेपकेस होता है. Which cell is the plant having rubisco? मेसोफिल सेल के अंदर यहां पर बंडल शीट में will get rubisco, right? Bundle sheet gets rubisco here. Can you see, students? Bundle sheet gets the rubisco enzyme here. Gets the rubisco enzyme. Okay. So no photorespiration in C4. No photorespiration here. Photorespiration नहीं होगा यहां पर C4 के अंदर. That's all you have to remember, students. That's all you have to remember. Main takeaway is that in C4 cycle there is no photorespiration. Now, what are the factors affecting photosynthesis, students? What are the factors? Now, there are something called as your internal factors and external factors. Internal factors are mostly dependent on genetics, number of leaf, type of leaf, pigmentation, chlorophyll concentration, है ना, size of a leaf, है ना, वो सब matter करता है. All of these are the different internal features. Now, what are we interested in? We are interested in the, students, we are interested in the external features right now, external features. Okay, so light, CO2, temperature and water. Now we will understand, students, a concept called as limiting factor. Ready, students? Ready? Okay. Now what is the meaning of limited factor or limiting factor? Limiting factor is a factor which is present in the least amount and it can influence the entire reaction. इसका मतलब क्या है स्टूडेंट? इसका मतलब वही बहुत ही सिंपल है. अगर सोचो आपके क्लास में ना, आपके क्लास में एक जो बच्चा है, एक बच्चे से एक स्टूडेंट से ना पूरे क्लास का नाम खराब हो सकता है ना, वन स्टूडेंट से. Now here also there are multiple factors here. Multiple factors are here. Out of these multiple factors, one factor is present in least amount. It can determine the entire rate of reaction. I'll give you a very quick example, students. Let's say it is very cold winter time. Now winter time के अंदर students, early morning, do we have sunlight? Students, tell me in the chat quickly. Winter के अंदर early morning time, early सुबह 4 बजे 5 बजे, do we get sunlight? Do plants get sunlight? नहीं मिलता sunlight. So can I tell in the early morning time the sunlight is a limiting factor, है ना? Because of the sunlight is very less that time. Sunlight is usually determining the entire rate of reaction. Now let's say अभी desert के अंदर तो सब हर जगह हर टाइम most of the time we have sunlight. Day time पे sunlight है. So will sunlight be limiting factor there? नहीं. So can anyone tell me out of these 4 different external factors here, in desert condition, which is a limiting factor? Anyone? All of you. In desert, what is a limiting factor then? In the case of desert, water is a limiting factor, students. Water is a limiting factor because पानी बहुत कम होता है ना. So whichever factor is present in least amount, it is called as limiting factor here because it can determine the entire reaction. ये देखो. If a chemical process is affected by more than one factor, यहाँ पर more than factor है, then its rate will be determined by the factor which is nearest to its minimal value. सबसे कम जो है ना, that will depend on that. It is a factor which directly affects the process if it is quantity is changed. So depending on the situation, we can change it. Now students, generally sunlight is not a limiting factor. Sunlight हर जगह होता है except the forest floors, except in dense forest ना, उस जगह पे थोड़ा sunlight कम होगा. So in that condition sunlight can be a limiting factor, but generally sunlight is never a limiting factor because sunlight हमेशा होता ही होता है sunlight. Okay, so please students, and remember that more sunlight is also very bad. ज़्यादा sunlight भी अच्छा नहीं है plant के लिए. Okay, because it can the pigment chlorophyll is there, no, it can undergo photo oxidation, destroy हो जाएगा pigment chlorophyll अगर ज़्यादा sunlight मिल गया तो भी. So sunlight is not a limiting factor never. Okay, now students, but what if I tell you the most important, the most ultimate limiting factor in plant is CO2. ठीक है, it is CO2 because students it is a major limiting factor क्योंकि हमारे atmosphere के अंदर there is only 0.02% of CO2 present, only 0.05. So 0.05%. Even if you increase it, students, even if you increase it by 0.2, massive change will happen. Okay, okay students, we have 0.02, right? So it is very important. So even increasing concentration up to 0.05% causes an increase in CO2 fixation rate. So we have 0. Something. Okay, so it is, students, it's very, very important, very important. Sunlight is a major limiting factor, students. Okay, sunlight is a major limiting factor. Okay, in the atmosphere we have 0.03 something. Okay, now both C4 plant and C3 plant both will get affected by CO2 concentration. But what if I tell you C3 plants can take more CO2, they can absorb more CO2, their saturation is beyond 450 microliter, but C4 plants just 360. So CO2 is beneficial for more CO2 can be taken by whom? By C3 plants. So that is why in the greenhouse where the CO2 is very more, we generally grow C3 plants. देखो इस greenhouse के अंदर ना, in this greenhouse we generally grow bell pepper, tomato, है ना, because they are C3 plants and C3 plants can take more and more CO2. They can take more and more CO2, students. Okay, tomatoes and bell pepper. Now temperature is a limiting factor. More temperature, less temperature, both are bad क्योंकि enzymes, all of these enzymes like peptase, right, Rubisco, all of these enzymes are very temperature sensitive. अगर temperature बहुत बढ़ गया, enzymes will denature. Temperature कम हो गया, enzymes will become inactive. So these enzymes are, temperature plays a very important role in limiting factor. Now the last one is, students, is water. Water is the last limiting factor here. Okay, now water has much more, if I would say, indirect role here. Indirect role. Now what do I mean indirect role, students? Okay, let's say the first condition, let's say there is less water. Let's say there is less water right now. तब क्या होगा? अगर पानी कम हो गया ना, will the leaf curl inwards? Will the leaf curl inwards? Yes, it will curl inwards. So there will be decrease in surface area. Decrease in surface area. If there is a decrease in surface area of the leaf itself, अगर हमारा this entire leaf will turn inwards और कुछ हो गया, surface area कम हो गया, तो surface area कम हो गया तो decrease in photosynthesis will happen then. Decrease in photosynthesis will happen, obviously. Okay, this is one. The second one is very simple, students. Let's say अगर पानी कम हो गया ना, अगर पानी कम हो गया, all the stomata will close. All the stomata will close. If stomata is closing, CO2 entering is very low. CO2 is very less entry. If CO2 is very less entry, then decrease in photosynthesis. That's all. Indirect role of your water as a limiting factor, students. Okay, students, with that the chapter is over. In just two lectures, we have finished the entire chapter. In just 2 lectures, can you believe, students? Okay, so don't lecture dekho. Next I'll teach you respiration in plants. In the next class I'll teach you what is Krebs cycle, what is glycolysis, and what is electron transport chain, what is alcoholic fermentation, what is lactic acid fermentation, in the next few coming classes. So we will finish the entire main, this is the main, right, entire plant physiology in the next week. फिनिश कर देंगे स्टूडेंट्स. उसके बाद आपको मैं एक पीडीएफ दूंगा द प्लांट हार्मोन्स के ऊपर. इफ यू लर्न दैट वन पीडीएफ, सारे के सारे प्लांट हार्मोन्स क्वेश्चंस यू कैन सॉल्व वेरी इजीली, और उससे ना हमारा पूरा का पूरा प्लांट फिजियोलॉजी खत्म हो जाएगा. ओके स्टूडेंट्स. सर ह्यूमन फिजियोलॉजी इज नॉट डन. ह्यूमन फिजियोलॉजी आई हैव डन 2 chapters. I will start with digestion next week. Next week I'll start digestion. I thought we'll take a break from human physiology and we'll start plant physiology. Plant physiology थोड़ा जल्दी खत्म हो जाएगा. Okay, human physiology 2 more chapters are left. 2 main chapters, digestion, excretion. ये दो बहुत इंपॉर्टेंट है. डाइजेशन, एक्सक्रीशन बहुत इंपॉर्टेंट. बॉडी फ्लूइड, respiration both are over. Okay, so thank you so much, students. Bye, all of you. Take care. Notes will be available in description. I will send it to Hardik sir. Hardik sir will upload the notes in the description. Okay, so thank you so much, students. Bye, all of you. Take care and have a good night.